what I propose, we can start now, because this morning we have to write some slides for the session of all groups together. So we had a sort of flavor of this session yesterday afternoon when we went to Group A. So I think we can just wrap down our discussions, and so I've taken some notes, but I'm sure that Cheyenne and Sandrine have taken good notes, but much better than mine. How do you want to proceed? Do you have some slides, or do you want that we write the slides down together?
I took a lot of notes, but I don't know what the best way is.
Yeah.
I think we can organize the slide around different titles and different parts, and we can then complete.
Or we can take the slide with the questions and write down the answers when corresponding to the questions, as you want. It's the simplest way, but perhaps it's a bit long. I don't know how long we have this afternoon. I think it's a discussion session. It's 1 hour. Or?
Yes, but should we organize the answer around common question with human question, because otherwise it would be very repetitive, I think, no?
Yes, it's a good idea, because there are some are very similar between the groups. Nell was out. I did not see. Please, could you let Nell or Romance enter, please?
Hey, I'm back. I was kicked out of the meeting. But I was still, I was working this morning on the VPN at the Institute. So I put down the VPN so that I'm on my own Wi-Fi. I think that's more stable. I hope that's more stable. Morning.
I'm opening my notes from yesterday. Does somebody from both of you, Sandrine and Cheyenne, do you want to share a PowerPoint slide, and we write, we do a live session of writing, or what do you think?
I could do, I don't mind doing that, yeah.
Yeah, okay.
Let's do this.
So you have to put it onto a script. So I borrowed it, and then you have to load it into your. They help you. So you've got a particular file for your talk, and you have to load your slides into it. Yeah, right down the end. Yeah.
So these are the questions, and then I took some notes, but I can get rid of my notes, and we can just add what we want.
I wouldn't throw them away.
Yes,
I have this saved
I
elsewhere.
agree. Because we can complete, but
Yeah.
it's interesting to have the
Maybe,
And,
and that's a real.
so these are just the the notes I took during our discussion for each set of questions.
Okay.
So it is pretty long. That's why.
Yeah. Maybe first to get this going, how much time do we have to present? And are we going to present? Then the second question is, are we going to present each question separately, or are we going to group a number of questions depending on the time that we have for giving feedback? Christelle, do you know?
So we were discussing that just before, to group some questions, because some can be similar into our set of questions, and moreover they can be similar to or complementary to other questions from the other groups.
Mhm.
So the questions were divided into comparative approaches, experimental designs, and then individual population ecosystems. So we could just stick to that maybe if it's
Yeah.
already organized like that.
Yeah, that's nice. Yeah.
So the
Do we
only
just-
last one is perhaps specific to our topic, but the two first one, comparative and experimental design, I think we kept the same structure for the other groups.
Yeah.
And what about the question of multi-generational and transgenerational? Do we only
The
focus
definitions.
Yeah.
no, but do we focus only on transgenerational because humans are only interested in that, or should we stress out this point mainly also? But
I think we shouldn't be obedient to the only human side point of view. We are there to present also the non-human biota. And for the non-human biota, the multiple exposure situation is more relevant. So I think we need to keep it.
yes, we need to keep it and stress, emphasize the difference, I think.
Yeah.
Probably.
I'm not sure they are so different, because yesterday we spoke of some specific situations which are not medical, where people may be exposed for several generations. In situ, for example, you have the Kishtim accident, where people are still exposed to high levels of contamination. You have the high background levels. So we can also speak, give our idea even for humans. It's not forbidden.
Like even uranium miners, they're exposed to dust and a lot of other hazards.
Mm-hmm.
And also for transgenerational effects, yesterday we said
do we just want to go through the individual questions, take notes, and then maybe we can combine into slides at the end? That might be the most efficient. So question one.
Sorry, I just have to leave for 5 minutes because I have to upload my presentation in another desk. So I will be back. Go on without me.
Yeah, what points do we want to make here? I think the point of the multiple versus trans is a very good one. What we could do, but I still, Christa has not said how much time we have to present this, but we could show the questions and then go to another slide and
so something like simple like this.
Yeah, so say that within comparative approaches, we had 4 questions. We don't read them aloud because that will take us too much time. And then I would go to the next slide saying that these are in general the discussion points that we had and the answers that we came up with, or something like that.
Discussion. I guess we could just say discussion. And then here we can go over our different points. Okay. So these are, I believe, I'll just double check.
I think we have no more than 15 minutes. I don't know exactly, but I am looking.
So yeah, these first 4 questions are comparative approaches, so they go to.
Yeah. And then the answers that we came up with, yeah, I think we can make a first point on the transgenerational versus multi-generational, saying that
I think that's for the next section, though. The next section is experimental designs. I think this one is more. This one's more focused on lab versus field studies, and maybe comparing animals to humans.
Yeah, and which model is that also there? With no, which model species is also?
Is there a best suited model organism to study?
Yeah.
Yes. Yeah. I liked our discussion about complementary. I think that was a very good point. That comparing.
Yeah, that's true.
Person. How to word this?
Rather than.
Yeah.
Complementarity then. Complementarity.
Complementarity. This sounds weird.
Or comparison. Yeah, no, it's comparisons provide complementarity.
Or provide opportunities. Or how did we say more complementary than comparative? Or can we just actually say that?
More complementary than comparative.
Complementary, then it's again complementary. And in that sense, they're needed in a way.
So we said some parallels to doing epidemiology. So do we like that point, that it's helpful for validation of things we discover in the lab?
For the field, yes, I think.
Yes.
I think it's needed. So fields.
Or we could just have a general point about field versus laboratory. Because what else did we say? Field, easier to study, multi-generational. So this was a point Christelle made, something after, say, an accident. If you go out to the field, you can get information on multiple generations at a single time versus generating them in your lab. I don't know if we want to emphasize that.
Lab is a controlled situation to discover the marker, and we can validate in situ.
Environmental relevant.
Yeah.
Yeah.
And then we said. Any other points you want to make for lab versus field?
I would say lab field studies offer an opportunity to validate findings in the lab and vice versa. So if you see some,
that's true, yeah. It's very true, yeah.
Yeah, maybe then No, actually, in the first point, so that the fact that we say lab allows for control experimental parameters, you already have that are sometimes missing in the field, like the time zero and a way to get rid of confounding factors. That is all possible in the lab of some of the confounding factors.
Allow for control of experimental parameters. Confounding factors versus field studies allow for understanding.
Yeah.
Of environmental relevance.
Yeah, that's okay.
Anything else we wanted to talk about?
No, maybe you can put on the first bullet point you have on the cross species. For me, on the fact that they're complementary and rather than comparative, that holds true for both cross species comparisons as for field and lab studies.
Yeah, that's true. So do we want to add a point here about them complementing each other, or were you mentioning for the first bullet point?
You could say cross species as well as lab to field comparisons are more complementary. And then the next two bullet points are actually a explanation on what we mean with that.
Yeah. So wait, cross species.
And then for the cross species, maybe we still need a bullet point.
So for cross species, these are all our discussion points.
My God, we talked a lot.
Yeah, it's. I tried my best to write down what we were talking about, so this bullet points.
It really was not obvious that you were writing down so much. You're a very good.
Is also variable for radio sensitivity. We talk about that.
Yes, that's true. Radio sensitivity and also
sensitive step
to be representative of each
trophic level and
level and environment compartment.
Yeah.
How do we want to phrase that when comparing different species? Important to consider differences in radio sensitivity. Or do we want to add a separate bullet point talking about that?
For me, once again, it's not really comparing. In fact, it's more
studying different species.
Yeah,
I like that, studying.
Yeah. In radio sensitivity, trophic condition and compartment media.
In habitats or
yes.
Yeah. Or scenarios.
Or can we say like no, not ecosystem.
How do you say it? No, it's gone.
In French, it's compartment, but ecological.
Yeah, and ecological compartments. Yeah, I don't know how you say it.
Compartment.
Also, to be representative of reproductive strategy, also. we talk about
Yeah,
that.
we talked about that too. Yeah, that's true. Yeah, the fact that, yeah,
And
for
genetic,
example,
clonal. Sexual, yeah.
and actually, you want to make sure that you cover with your studies, that you cover the different strategies, because you don't know whether the different strategies do not result. If you only look at sexual reproduction, for example, you can miss out the fact that clonal organisms might be more sensitive to radiation. Yeah, that is actually why you need to study the different species and the different situations, both the ones that are more sensitive versus the ones that are also the ones within the trophic levels and so on. I think that was the point that we wanted to make.
Yeah. How do we want to phrase that so it's not too long? Because it's not just reproductive strategies, right? You know, there's a lot of differences between organisms. I think we focus on the reproductive strategies, obviously, for this task group, but
yeah,
I think it's important to consider just different biota in general. I think
yeah,
that's what we want to say.
Because
Maybe
also life cycle duration is really important.
yeah,
like diversity. Maybe we could just say diversity of biota is critical. Just so then again, you're considering different aspects of the food chain, different players in the ecosystem. Maybe we just keep it simple like that. And if Christelle's presenting, she can just kind of emphasize the reproduction and what we talked about.
Yeah.
And we were talking also about if you want to protect the ecosystem, about the key species of the ecosystem.
Yeah.
We don't have this. Yes, diversity of both is critical, and key species are critical in ecosystem.
Yeah.
Yeah. I think that would go under. Maybe we can have, or we'll start a bullet point, but we can make a separate slide if needed. So for best model organism, this is what we talked about, it looks like.
Yes, yes, okay.
I think our real conclusion was this, that there is no real single best model.
Yeah. Yeah.
Yes. No single best model and no single approach. Because everything is necessary to be complementary.
Yeah, that's an important one.
Because we talked about, you know, something lower on the food chain that others rely on. So that's an important point.
Yeah. And that's also what I wanted to say by showing yesterday evening the slide on the interaction with the of the butterflies with their food source. If you you can see if the food source was not affected by the radiation, which was no problem for the plants growing there, then the butterflies would not have shown the morphological abnormalities. So
we said today that the model should fit the purpose that we have. I think it's important to have that because it's in common with the other group.
Yeah, it should. Your model organism should be chosen wisely in order to be fit for purpose.
How do we phrase that? Model organism.
a wise choice of setup will deliver most relevant information for your set out goals or something.
Yeah, that specific. Yeah. It's always about context, right? Like what endpoint, what that's why it's so hard to generalize these things. Should we just say specific, maybe not outcome, but
specific question or scientific question you want
for testing your hypothesis?
Yeah, for testing.
Meaning that you are doing research that is hypothesis driven.
Yeah.
It's always what we do, huh?
Yeah.
It's split this one.
Yeah, I think it's better to split it into.
This. Is there anything else we want to talk about for best model organism?
Yeah, maybe on the discussion that we had with Group C, to see because they were asking for a best model organism for humans.
What did we talk about there? This is what we discussed.
I think
go, Sandrine.
C. elegans and zebrafish are already used for toxicological experiments, so it depends. It's always the same. It depends on the question, and I think we need to adapt the model to the question. So,
do we want to make the comment about very common organisms like C. elegans, zebrafish, fruit flies that have been characterized so well that it allows you to do maybe more omics or more precise biology? So there is benefit in them, but I agree Sandrine's point. We shouldn't only focus on zebrafish, right? They're not the only fish that are out there. So it's a balance of using the tools.
They don't even swim most waters because they're very specific to tropical environments.
Yeah, I think it's already known that such models very characterized are very interesting for toxicological point of view. And Christelle told also that C. elegans have had 2 Nobel Prize and did a lot, make a lot of science. Yes, for medical purpose. And so I think it's important to
maybe add
to notify that
there to have a plant.
Yeah.
Yes.
Dopsis, did I spell that correctly?
Yeah.
Because among the groups you have also epidemiologists and dosimetrists, so it's worth repeating things.
Yeah, you're right.
As well as known sensitivity to toxic and stress responses. The fact that a lot of these organisms have been used in ecotoxicological testings also makes them very valuable, because you know about how you do a setup, how you test for toxicity, what are the sensitive endpoints. There's a lot that you can not that you have to name it here, but that's the background here.
Pathway are simpler, and everything is you don't have any many organs, and it's a simplification of the system. So I know that all that point are new, but are known. Sorry,
are known, but maybe not to epidemiologists.
Yes, you're right. You're right.
What about something like environmental sensitivity? That's a more general term.
Yeah, that's true.
And also, I think that we can say that there is no single best model also for human part, because it's something that they discussed yesterday. Huh? They are also searching for.
So I think we could combine this point here.
Yeah.
This is for population. So I think this point was that we have to consider the actual environment, real ecosystem. So I think that point.
Yeah.
Nice. Do we want to make a point about humans specifically, since that came up with the other group?
But perhaps not state down that for them, because perhaps it's not. But we discussed about thinking about a theoretical base model that could help to define this base model, and then look if we have it. So it's something that is common with the human approach.
Yeah.
But even for us, if we had to do that, even to find the theoretically best model.
Yeah. Any other points for model species we wanted to write down? Do we want to, I think part of our discussion was specifically if we are interested in transgenerational effects.
Yeah, but that fits more on the second part where we look at the experimental setups.
Okay, that's fair. Yeah.
But why did we remove the part on reproduction?
It's still here. This slide, I just split the slide so it's a little cleaner.
Because.
So I think the plan was these are the 4 questions for comparative approaches, and then part of the discussion was just comparison across species. Part of the discussion was about best model organism.
Maybe for the first slide, slide 4, it's cross species as well as lab versus field.
Yeah.
So
I think
that needs to be in the title. Yeah.
We can put approach comparison, no? Cross species and.
This.
Right.
Yeah. Okay. Then you combine actually the 2 first questions.
Yeah. Because number 1 was field versus lab, 2 was cross species, 3 was best model, 4 was why are transitional effects observed in biota and non-humans.
Yeah, I think that we said we're not sure about the fact that they are not present in humans. It's harder to see because there are.
I think it was harder
because of their expected lifespan being advanced and things like that.
And also medicine.
Yeah.
I think we were also saying that the challenge is to be able to definitively prove that it was the radiation that caused it is what's challenging because of all of these other parameters.
Yeah.
Do we want to just make a slide on that?
I don't know.
It was more of a minor point that we discussed. I don't know, Crystal, if you think we want to make a separate slide for that.
We can make a separate slide for the moment. I think that we have 1 hour and a half for the 4 working groups, but including the discussion together, and including also discussion on the special issue. So I think we have
20 minutes.
20 minutes.
no,
Yeah.
I would say 15 at the
15
maximum.
maybe.
Yes, 15, not more.
So 15 slides. It's okay, 1 slide per question.
So what do we want to, how do we want to phrase this slide?
Yeah, I think the first point is actually that the endpoints that we look at in non-human biota versus humans is already quite different. So so you look for humans mainly to stochastic effects like cancer, which are hard to, and then you can make that definitely state that radiation exposure in the first generation resulted in the
Do we want to just say
fecundity, reproduction? Yeah.
Reproduction, growth, and survival.
Yeah. And population survival.
Yeah.
In fact, in biota, we are looking for endpoints that are more integrative than in humans.
Yep.
Because in humans, you are looking for one individual, what happened to one individual, and in biota, we are looking population level.
Yeah, that's why I said population survival. But maybe it's a good point to put it here as a separate bullet point. Yeah.
What's that sound?
Yeah, that's great. And then I would make the point for the transgenerational effects that it's hard to
prove
for the one that you say the on the cancer.
Is that okay? I don't know if that's too general. Your outcome with
Exposure in the first generation.
that occurred.
with parental exposure.
And it's not just parents, right? It could be parents, grandparents, and it keeps going up. I don't know if there's a way to phrase that. And or, yeah, we could just say, you know, parents, grandparents, etc. Like, I don't know if that's clear or if that's confusing.
Yeah, maybe to start that sentence, you can say due to the
multifactorial parameter.
Yes, stochastic. I wanted to say due to the fact that you look at stochastic effects due to the lifestyle of humans and the medical intervenence is not the one that I want to say, but the fact that we have access to medicine, there is difficulty. That is actually the 3 things. So actually, we cannot exclude transgenerational effects, but it's very hard to pinpoint them due to the fact that we have a very different lifestyle. So that's also the fact that we talked. I think we talked about food habits and stuff like that that might also differ a lot between the different humans, and therefore make it very difficult to really pinpoint that. Something.
So, mobility.
Yeah. The lifestyle, the mobility, the access to medicine.
Sorry, what was
factors?
Mobility, lifestyle, mobility, access to medicine.
And Christelle had something, but I didn't hear.
No, I said confounding factors in general.
Yes, that's true.
Yeah.
That's true.
Yeah.
And a lot of confounding factors, yeah. Numerous, that's a good word.
We have an English lesson at the same time.
What about that? Maybe is that and better?
Yeah. Yeah.
Okay.
Yeah.
Okay.
But,
are we happy?
Yeah, maybe we just have to put there, but we cannot exclude them because that is the most important, I think, for radiation protection, that we have to keep in mind that these transgenerational effects are there. So you want to protect humans also from it, but it's hard to pinpoint them in as such. But yeah, if they would pop up or be more prominent, yeah, then you need to be sure that your radiation protection system is appropriate.
So could that be maybe captured by this point? I think part of this discussion was specifically why are we able to see it in biota, and
yeah,
I think it's because you're designing studies that look at specific phenotypes or outcomes, whereas in humans, it's usually you're casting a very wide net, looking for very specific or not very specific markers, just changes across the genome, right? I don't know if we want to emphasize that.
It's your first point. Endpoints.
Oh, actually, yeah, we have that.
Yeah, I just wanted to add that
yes,
it's not because it's difficult to see them that they are not there. So
yeah,
therefore we need to, that is the point that I wanted to make. So imagine that they become more prominent, then it's needed to have your radiation protection system adapted to it.
Yeah. Something like this, maybe.
Yeah.
So important.
I was just thinking from an ICRP point of view that is why that we want to make sure that they keep on taking them into account, not just say, "Oh, we cannot see them, so they're not there," because the question was formulated as if transgenerational effects only occur in biota, which is I think. Is possibly not true. We are not different. We just behave differently.
Yeah. And we are exposed to different levels of radioactivity also, which is different from the biota, which is exposed chronically.
Yeah.
But anyway, we have to prove or to demonstrate that there is no transgenerational effects in humans. Not we, but them.
What about this? Still important.
Yeah.
For us, it's easy. We just ask the question.
But I agree with Nell. I think it's important to raise because otherwise others may just say, okay, well, if they're not there, why are we spending money and time? Should we just ignore it? But I think we're just trying to highlight that it's very difficult, but it's still important to consider.
Yeah. Yeah.
Anything else we want to talk about for the first set of questions?
Nope.
So that was questions 1 through 4. My notes. We can clean up the slides. So these are the next questions.
Yeah, they were very long.
Yeah, they're very long.
Maybe we have to
Split.
clean them up a little bit and put the, for examples, out of it.
Yeah, maybe study designs and approach it. Do it with this.
You can delete everything. Well,
Should we keep the last line about life stages or get rid of that?
No, maybe you can leave that.
I think it's included. Huh?
It can be part of the answer. I don't remember what we discussed yesterday, but
Where's question 8? Oh. So yeah, 5. I'll just double check. 5 through 9. The second set.
De onderste juist.
Yeah, 5. These questions were experimental designs. So maybe a slide on overall study design approaches, a slide talking about dose and dose rate, maybe.
there was also the discussion on the definition, so it can.
Yeah.
This is what I have for these questions. Yeah. Do we maybe want a slide with just simply
the definitions?
Yeah.
Yeah.
Yeah, I think it's important.
Okay. Is there a hyphen for transgenerational? No. Any other terms we want to define?
I think it's.
No, I think that's where the
you have heritable effect also, but
yes,
yeah.
I was thinking about heredity.
But for me,
did we really come up with a definition there? I don't remember.
Talked about epigenetic inheritance during Cisco's
that's
discussion. But I don't know if we actually defined it though.
No, but,
okay, it's transgenerational. Do we want to say effects? Or are we talking about transgenerational exposure?
Exposure, I think.
Exposure, actually, we're talking about.
So let me ask a hypothetical question. If I expose F0, I expose the parent preconceptually, and then I'm looking at, say, F3, versus I expose F0, I wait 2 generations, I then irradiate F3 or F2, and then I look at something in F5. Does it have to only be irradiation of F0 and then looking at effects F2 plus, or can it be irradiations across multiple generations? They just can't be in order, if my question makes sense.
Yeah, it's a very difficult one. It's actually in between. You what you look there for is priming. So you look at whether an historical exposure somehow protects or provides the organism with a different set of background so that it responds differently to the exposure. For me, yeah, it's difficult to say. It doesn't fit multi-generational. In my multi-generational for me is really each generation having the same kind of exposure. So it would fit more in a transgenerational setup, where you want to
I think
yeah,
the
but
key for transgenerational is you take the exposure away at some point,
It's
at
right?
some point,
Combination, yes.
yeah.
So we said exposure of the parent only.
Okay.
I don't know if we want to say difficult to see and feel. Get rid of that point. So for multi-generational, so I think the key we talked about for multi-generational is that it's the same pattern. It doesn't have to be continuous. It can be acute or fractionated, but the key is that it's consistent across the generations. You can't, fractionate it one generation, then change it to a different type of radiation. Like we're saying, it has to be consistent.
Yeah.
Yeah. Same radiation pattern.
Not acute, we said chronic, no?
We're saying it could be acute, no? Wasn't that? Maybe I'm confusing our discussion.
For me,
like let's say
it's not only continuous on all life cycle, on all life lifespan, but it should have a notion of a quite long period or developmental stage or acute, repeated. I'm not sure that it's multi-generational, but it's my.
So if I took my parent mouse F0, gave it 1 gray, F1 gave it exactly 1 gray at one time point, F2 1 gray only. Is that multi-generational by our definition or no?
It depends 1 gray on how many time.
Once, or just say once, at a very specific time point that I'm interested in, I'm exposing it the exact same time point every generation.
You can study.
If it's acute 1 gray for me, sorry, excuse me, Christelle.
I think you can study multi-generational exposure for acute or chronic pattern of exposure. It depends on your question. If you, for example, if you want to compare the effect of multi-generational exposure for 1 gray of acute or chronic exposure, it's still possible. It's perhaps less adapted to the environment, but written like that, it can also be adapted to medical irradiation. It's multi-generational exposure. It's more over the time pattern than the irradiation. What do you think?
But I don't understand in which kind of scenario you can have that, except medical, okay? But it seems.
If you consider nuclear power plants, so
yes, release,
acute, but not high levels. But when they release the primary waste, waste, it's an acute pattern.
It's kind of like seasonal, right? It's acute, it goes away, and then periodically it keeps coming back.
Yeah.
So how do we want to phrase that? I think the key is that the pattern has to be consistent, right? You can't do acute one generation, then change it to a different, was kind of what we were saying.
Perhaps the term acute is a bit sharp. Could we say transient or chronic?
Yeah, I like transient, yeah.
Yeah.
Because in the environment, it's rare to get very acute kind of stressors, right? Usually they are persistent and they stick around, yeah. I like transient.
Yeah, we also said seasonal.
Yeah, seasonal. It's okay.
Do we want to add that, like transient, seasonal, or chronic?
Yeah.
Yeah, okay.
So same pattern of exposure.
for each generation, actually.
Do we want to make this point about difficulties to observe in the field?
I wouldn't put it here.
I guess this
Maybe-
is the definition.
Yeah.
I like this point. We talked about the recovery. I don't know if it fits here, but I really like that point of discussion for environmental protection. I think this is a good point to raise at some
Yeah.
point. Anything else for this slide, or are we happy with these two definitions then?
I'm happy.
Okay.
So how do we want to talk about study designs then?
Don't know whether we said much about study designs.
We talked about lab and field already, so.
Yeah.
Oh, I think the study designs was kind of the omics. So I had a little table that I put our main points down.
Yeah. Yeah, I think it's what we did say, that it was important to go across different levels of biological complexity, because you want to have the link between the omics and the molecular ones, and as well to the adverse outcome. That is something that we said. So important to go across.
Oh, we look at multiple levels.
Yeah, that we said.
Multiple biological level.
Important to consider.
Yeah, they say levels of biological complexity is actually the term that they use, from molecular up to individual or even population.
So we're saying like cells, the molecules, the tissue, organism, population.
Yeah, the molecules are. It's
Before.
actually it's
Like-
it's actually
Like just this?
molecular, and then
Biological pathway.
yeah.
What's
The molecules are in the cells, so
Yeah, like
it's
molecules in
subcellular, cellular, tissue, whole organism. So maybe
And
at
population.
subcellular, then you have, but then that comes before cells.
So subcellular would mean like at the level of Ross, like all of those things.
Yeah, the molecular level. Yeah. And tissue, maybe you can say organs.
Yeah, tissue or organs.
Yeah, so you compare a leaf or a heart or a brain or.
Yeah. You could look at whole organ systems as well.
Yeah.
I feel like this is really an AOP. Like that's all this point is getting across. So
Yeah.
So by that argument, should we mention that you need to study different endpoints? I think it's not appropriate to just pick one. It's probably more appropriate to look at. I don't like to keep using the word important for every bullet.
Analyze.
Analyze different, just say analyzing different.
But I think once again, it depends of the objective and the question.
Yeah.
Because,
Yeah.
are we obliged to to study all biological levels. I'm not sure for radiological protection. The most important probably for radiological protection is a final endpoint that is the adverse outcome and the population level. So it's important to have different endpoints at different biological level to explain how does it.
Yeah, that's true.
Anything else we want to talk about? I don't know what else to say. Because I think this question, a lot of the discussion was mainly around phenotypic endpoints versus omics and molecular kind of endpoints.
Yeah. What was in your table? You had a nice table.
These are just kind of my thoughts I put together. Where to go?
Slide 6, I think.
Oh,
yeah.
I think this is always what you have to consider. Like for field studies, carrying a lab scale to measure your animal versus trying to carry a flow cytometer.
This table is interesting.
Yeah, we just put the table there. It's a nice summary.
Do we want to change anything or add anything?
No, I think but you perhaps it's not. We want to read it from the first line to the last line, but there are
a number of good points there. Yeah.
Okay. Maybe just combine.
Yeah, I think you can then for your bullet points, the bullet points, multiple endpoints. It's I think it's in there somewhere. No, it's not. But
no.
But that's quite obvious if you say that you are going. Yeah. So maybe that one can go.
Okay. What about consider different endpoints?
Yeah.
And levels of biological.
Yeah.
So that way we can
just that then. Yeah.
Get rid of this.
And then you have your important in front of your endpoint.
I think it is a good point to emphasize though, because I think in humans, we always talk about biomarkers, right? Can we find this kind of golden standard? But it's almost impossible in the real world. So I think it is important to emphasize different endpoints so you can get different parts of the story, right? And that we talked about tissues yesterday with the human group, and my argument was we should consider every tissue. So okay,
it's good.
Okay. What other questions do we have?
We must be careful to the time because coffee break is normally at 10:30, but we can skip the coffee break. But
I have a short meeting during the coffee break, so I need my my coffee break for not for having coffee, but for doing something else.
So I didn't have too many notes for the other questions. This is all I wrote down. But I think part of our discussion was about endpoints and facts that we could use known ecotoxicology to guide our choice of endpoints, right? It's not that we have to develop these from scratch, but we can use kind of toxicology and other stressor models to our advantage.
Yeah, maybe we have to add that to the slide somewhere. That we, I remember that we said that we don't have to invent the wheel again.
Yes, the point about chemistry and so on.
Yeah.
Chemicals, we talk about this.
It's hard to put it there. So you need but it's hard also to put it on a completely only if that's the only point we want to.
Well, there's that, and then our discussion of dose rate. these were my original notes, but I didn't write anything down for question 8. But I think our kind of consensus was you have to consider dose rate, and time of exposure was.
Yeah.
I think we say for dose rate, we say that it's not specific question for transgenerational exposure, so we can probably skip it, no?
Yeah, that's true. We said that.
Yes, but there is also the question of the historical dose.
Yes,
Yeah.
that's true, but
Maybe just a slide. We don't have to make it super long, but
it's not totally the same that the.
So what points do we want to make here?
We say that both.
But we are talking about cumulative dose over generation, something like that.
I think
Are important.
Finding absorbed dose. Like, how do we want to phrase that?
Yeah, that's a difficult one.
I don't know.
We also discuss more generally about the importance to report dose rate, time of exposure, and duration of exposure, and life stage.
So I think it's important to mention that both are important with duration time and life stage also. We talk about life stage exposure.
Developmental period of exposure, or stage or life stage of exposure, because it may not be just during development, right? It may be kind of later in life
it's
as
okay,
well.
developmental period of exposure.
And then what else? Dose rate time. Or is it just those 3?
And duration of exposure also, which is
and the recovery maybe.
Duration.
Or length of exposure, I don't know.
Duration is okay.
Yeah. And then do we want to comment about quantifying dose? Because I think that is probably important for any protracted exposure, right? How do you accurately define the dose? Because there's different ways to look at it too. You can construct dose to certain developmental time points. You could just look at total across their total life, how much they accumulated.
We did not discuss about that, but for the environment, we only speak about gray. We don't have any.
Yes,
yeah, we don't have sieverts.
Yeah.
So perhaps just say that dose quantification is already complicated due to several.
Yeah, it's actually you need to do a good dosimetry, but it is indeed true that we don't look at the impact on the tissue in the same way as we don't take into account.
So does this point capture it then?
Yeah.
Wait, sievert is equivalent dose. Oh wait, effective doses. Okay, yeah, it is equivalent.
Yeah.
So what's the second bullet point we wanted to make?
It's more those
reconstruction
for we are in transgenerational exposure.
I think that's an important question, is for multi-generational specifically.
Yeah.
How do you define dose? The dose, every single generation you just start accumulating? Is it only within a generation? I don't know if we really discussed that in detail, but that's probably an important thing we should discuss if we're talking about multiple generations. Because normally for humans, we talk about within one lifespan, right? It's rare for you to talk about accumulating dose over multiple generations like that.
Yeah.
So dose or
for all exposed generation or something across generations.
Generations are exposed.
Reconstruction across generations, something like that.
Yeah, I think it's okay.
Across. Important when studying multi-generational effects. Because I think for transgenerational, by definition, it's easy, right? It happened in one generation, and then it's taken away, so there is no more exposure. So we can say multi-generational here.
Yeah, but for recovery, if you want, you cannot consider only the present dose, because it's recovery, and if we suppose that there is an effect from parental, you need to consider that. And so how do you that? For me, the problem, the issue is the same for multi and transgenerational, because for both, the historic dose is important. To my mind, I
like quantifying historical dose, like, is that how we want to phrase it?
For me, yes. It's, taking into account the historical dose.
Yeah.
Because I think historical dose makes sense, right? When people see that word, I don't think that's confusing at all. Okay, are we happy with that, or any other changes?
Yeah, I think it's okay for me.
Okay for me.
Okay. So for experimental designs, we have definitions, study design, dose versus dose rates. Anything else for this block of questions?
We had some discussion on hormonal.
I liked our discussion about the oysters. That was really cool, and I think it's really interesting. I don't know if it fits into this discussion. This might be more kind of very specific to our task group. I don't believe there's too much focus on hormonal crosstalk in humans.
It might be important to add a section if we have to write our report on this, that we added there, but maybe it's indeed it's not necessary for the whole group I think.
Okay, so are we okay with this then? So this slide is these 4 questions or 5 questions.
Yeah.
Our definitions, study design. Then obviously, like, you don't have to go into every detail in the table, but just say, this table is just highlighting kind of the advantages and key differences between the technologies.
Yeah.
Dose versus dose rate. Then, okay, so last block of questions. So I found this paper. Funny enough, Cisco talked about almost all of these points. That's what that was.
Can you send the link of the paper? That would be nice.
Yep. I can put it in the chat.
Just in the chat.
How do I open the chat? I really like this one because they kind of identify specific points, or explaining how epigenetics influences. And like I said, we talked about, I think, pretty much all of these. Just leave them up. I don't know if we want to go into this level of detail for this. I think it is an interesting discussion, even though it's not really, we don't talk about human evolution in this way. I feel like human the human people could be interested in something like this. But again, it is kind of more specific for non-humans.
Yeah.
But I think there is a point to bring up for human protection is if theoretically this could be happening in humans, we need to justify studying these in humans because, again, it's the paradigm of I go get a CT scan and I'm worrying about my great-great-grandchildren, right? It's a kind of bizarre scenario, but we're talking about those kinds of implications.
Yeah, most of the
want to say
times
that.
we are less people are less altruistic than that. They're more egocentric oriented.
I don't want to make that point. Yeah,
no.
I'm just saying, like, theoretically, I think these kinds of questions are valuable. I don't know if the human people will care too much about evolution.
Yeah.
I think they have the same question in their group, and perhaps the question of imprinting subsequent generations can be important for them.
Mm-hmm.
Perhaps you can keep that one and highlight only the point 5 and 6. I don't know. These 2 points are maybe general for human and
yeah. Do we want to keep all of the points or just maybe highlight a few and just say, you know, from this paper, these are a few that we thought were of specific interest?
Yes, perhaps only keep the two ones that will be commented.
I don't know if it's in this group or during lunch, we talk about to study for transgenerational effect, mainly DNA methylation, because this is the most stable modification you could have, and could be good in first time to focus on DNA methylation instead of small RNA or stuff like this are less stables. I don't know if we discussed this in this group or it was another discussion.
We had a little bit of discussion on it, and I don't fully agree with the fact that the DNA methylation is the most stable one. I think histone modifications are also there, although we also said that some histones are replaced during the reproduction. What we did say is that micro or non-coding RNAs are more rapid and therefore regulate gene expression more on a
dynamic. We're talking small dynamics and DNA methylation. It's a quick response to a stress. We talk about this or not?
Yeah, that we did. That we talked about.
Non-coding RNA is also tricky because they sometimes have multiple targets.
Yeah.
And those targets have different transcription factors affect different genes, so it gets complicated very fast for sure.
Yeah.
So I guess we can do this, or this again. I just kind of added. I don't know if we like this summary diagram I put together, but this is kind of how I picture everything interacting. I don't know if it's the most appropriate or not.
It's nice. We could think about adding that also, some kind of scheme like that in the report somehow, which is always nice to have a figure, not to.
Okay, so what?
Epigenetics and evolution. Yeah.
Then what do we want to call the next slide? Because this block of questions is called Implications at Individual Population and Ecosystem Level.
Yeah, we talked about the it's again importance, the importance of to look at ecological interactions that we talked about yesterday afternoon.
Yeah, ecologically relevant transgenerational effects.
Yeah.
We said we want to define ecological relevance or just kind of state that it depends
You found a good definition, I think, yesterday afternoon.
Did we?
You
wrote it down. It's not in. I think it's in your.
Okay.
Not?
I don't know. This was for humans. I don't think it would be up here.
Yeah, I just talked away. I didn't write much down.
Crap.
We wrote something on phenotypic confirmation of molecular changes, but I don't know if it's enough ecological.
I guess that was part of the discussion. My argument is that if it doesn't translate to phenotypic change, is it really a concern for the environment? Is my argument. I think we were discussing, the strength of evidence where they show, the initiating exposure to a functional change, linking it to some kind of structural or phenotypic changes, the best evidence we can provide.
Yeah, we also talked, I'm looking back into my notes, we talked about the cost of adaptation.
Yeah, for me, it's very important.
Yeah, so it might be that the radiation on its own doesn't give a big of an adverse outcome, but the fact that you have to invest in adapting yourself to that unfavorable but therefore not harmful situation might make you more vulnerable to other stressors, which is something that is that maybe is also needed to consider to see how much that cost is. And that's it, that I wrote down as an important point. And it also fits under the fact that ecological relevant, because ecological relevant for me is taking into account the fact that an organism is not just living in an ideal lab world. It's being triggered by a number of things like food shortage or other environmental cues or and stuff like that. So.
I think this is where multiple stressors comes in.
Yeah, that's true. Yeah.
Yeah.
And in ecosystem, you have all this story with predators. If you have the predators, prey, and something like it's very important in ecosystem to food chain. Ecosystem.
Because that's where something like growth becomes important, right? If an animal is supposed to reach a certain body size and it's either bigger or smaller than that, both can have implications for predation.
Yep.
You can't like all these kinds of factors, right? But our discussion was that is very species and context specific. One species to the next, that entire strategy could change.
Yeah.
Anything else we want to write down? do we want to make a point about how many generations should be studied? Should multi-generational exposure or only parental exposure be considered? Because that came up during Group A's discussion as well, I think.
I wouldn't make it as a point for the whole group. I think we already had we define multi-generational, or do we want to make the point here that in the field multi-generation is more common than transgenerational exposures? I don't know.
This question was largely discussed yesterday with the A group.
Yeah.
For us, perhaps we can distinguish viviparous and oviparous species, where transgenerational effects are seen from third generation versus second.
So should this
it can be added on the definition, no?
Yeah, but
in the definition of multi-G and trans-G, no?
Yes, not here on the here.
We can specify here, no?
Yeah. So are we adding it to the multi?
Yes, for none on the cells in transgenerational exposure, you can put depending on model F2 or F3, no?
Something like that, maybe.
Okay.
For me, it's okay. But...
Okay.
Yeah.
Anything else we want to bring up here?
A title?
Oh yeah, probably need that. What was this?
Maybe put the ecological relevant transgenerational effects as the title, and then then you can put all the bullet points one.
Oh yeah, you're right. Okay.
We can say ecological relevance of transgenerational or multi-generational is the same, or we want to focus on
trans/multi.
Yeah.
Because it's true for both.
Yeah.
Yeah, that's good.
How many?
I think we have the main. Looking at the. I think if we already have with these slides a lot of information to put forward on 15 minutes, so
we could end it there then. Yeah, we don't have to add anything.
No. What do you think, Christelle?
Yes, I think 13 slides, it's sufficient, largely. Well, it's even a lot, but we don't count the first one.
What do we want to title this slide deck? What's our group called?
Group C. Transgenerational and non-human biota.
Yeah.
On biota, just I don't like the term non-human.
This is what it says on the program, so we should probably keep this.
You can say report from Group C.
Summary report, or something like that.
And once again, put multi also with trans, no?
Yeah.
Our title is transgenerational.
Yeah, I know, but
I would put multi in it.
Yeah, I like. That's true. transgenerational and multi-generational.
Yeah.
Great. Well, very good. We worked a lot.
I think we earned our coffee break today.
Yes.
Do you want to do it, Cheyenne? Because you are belonging to the TG working group.
I don't mind. Yeah,
You
we
want-
can.
Yeah?
Yeah.
Okay. Super.